Please be aware that at the 14-minute and 30-second mark, there is 3 minutes of poor audio quality. It is quite noticeable, but as mentioned, it's only 3 minutes, so you'll get through it quickly. Thanks, guys. Hope you enjoy this episode. We have a firefighter on again after Ryan Lair was on, but today's probably not going to be too much about firefighting. But he's similar to Ryan. He got into the New South Wales Fire and Rescue at a very young age, just 19. He has famously competed at the CrossFit Regionals in Wollongong one year for the Creature team. But now he is currently doing his PhD at the Laboratory of Aging at the University of New South Wales. And actually one of his supervisors might be a name that you've heard of before. It's David Sinclair, who's quite prominent right now in the field of aging. So, Tim Chalmers, welcome on the show.
Thanks for having me.
Thank you so much for coming on. So, being that today's topic is aging, I feel that I'm not too rude by asking, how old are you now?
32.
32. So you're a very similar age to me.
Yep.
So we're born in the 80s.
Yep.
How long do you think we're going to live bar any trauma or accidents like that?
Oh, hard question straight off the bat. I'm trying to get me on record. First podcast.
Well, I'm super curious because having listened to a fair bit of David Sinclair, it sounds like he thinks we're going to live for quite a while.
Yeah. It's interesting. It's really hard to predict. Based on the advances in medical science at the moment, the current trajectory, we're pushing 80 for average lifespan at the moment in most developed nations. So that'll sort of continue to go on. Maybe some of the cardiovascular disease, obesity sort of stuff is sort of put a little bit of a hold on that. But we'll still continue to push that out. The thing is there's a lot of convergent technologies that are coming together at the same time that's going to be hard to predict what's going to happen.
It's exciting,
So aging research in general, sort of basic science into aging is really starting to take off. There's more money in the field at the moment. Some really exciting things happening. There's a few clinical trials, not necessarily measuring aging as an outcome, but using diseases of aging as their outcome and sort of measuring, using basic supplements or drugs, really sort of monotherapy stuff that are seem to be getting some decent results that have shown really, really good results in animal models. So that's one side of it. And then you've got genetic engineering, which is a completely different side of it. And that's really taking off. If people have heard about CRISPR, there's some therapies sort of coming to the market at the moment for sort of single use cases for eye diseases or diseases where we know what the mutation is that we can change with a CRISPR sort of gene therapy.
Is this where they have been regenerating eyesight in mouse?
Okay, so that's
is that different again?
That's different again. It sort of is a gene therapy. So that actually came out of David's lab. So that was to speak about that for a bit. So there was a guy, Yamanaka, who got a Nobel Prize, and you've probably heard of induced pluripotent stem cells before. So this is where you can take sort of any cell in the body and turn it into a stem cell, so then you can differentiate that into another cell type. And this guy, Yamanaka, worked out that there was these 4 factors that's all you had to, you just take the cells and you deliver these 4 transcription factors. And this was able to turn this into a stem cell, and then you can sort of coax it into being this other cell type. What the student in David's lab did, his PhD student, he, instead of using 4 of those factors, he worked out that you might only need 3 of them. So one of those, they're called the OSKM. So for different transcription factors, it's like OCT4, SOX2. I can't remember what the K is, but M is CMYC, anyway. and CMYC is sort of an oncogene, so it can cause cancer. So he left that gene out. And what they did in a mice model, they went in and they crushed the optic nerve right, in these mice behind the eye, and macular degeneration and eye loss is sort of associated with aging.
Mm-hmm.
So they were able to sort of induce that just by sort of crushing the optic nerve. And then they went in and they delivered these three factors. So it was basically a gene therapy where you put those genes into a virus and then you deliver that virus to the eye and let the sort of the genes be turned on in that eye. And they showed that it was able to regenerate the optic nerve.
So the
And
eyesight
came back. Like incredible stuff. So
Insane, eh?
that's actually a study that's only really, really recent. They released that middle of last year or late last year. So that's like one side of things. That's one use case in the eye. But you know, if we can do that in a number of different tissues or like this
Body
sort of
parts.
thing, yeah. I mean, like if you're able to regenerate the liver, you know, which is such a sort of central organ in metabolism and sort of everything goes through the liver before it goes everywhere else. So there's some really, really exciting things in genetic engineering. And then couple that with the explosion in genetic sequencing technologies at the moment, where that's becoming super, super cheap, much more high throughput, much greater resolution of what's going on at the level of DNA. That's to the point now, whereas before you have these machines that are worth $1 million, $500,000 or whatever, you have a sequencing facility, you send your samples off and they come back. It's at the point now where we've actually got in our lab a USB plug-in genetic sequencer. It's cost
at your uni,
$1,000 US. It's at my lab bench, and I'll be using it tomorrow. Like that's where that sort of technology is going.
Yeah.
So
incredible price drop, right?
Yeah. I mean, you've probably heard of Moore's law, where you know the transistors and the electronics is sort of exponential, right, in growth. Every 2 years or whatever, it reduces by half in price. Genetic technologies is far exceeded Moore's law.
Wow.
Like far exceeded.
That's incredible.
It's crazy. Yeah, it's crazy. So, and then couple that again. So these are like three things, right, that are going on at the moment in aging. AI and like big data science, machine learning, and this sort of stuff. That is likely to change all of science in general. So
quantum computing.
Yeah, like just the amount of iteration that you can have through data sets and sort of get to a result quicker. It's really, really hard to predict, you know, like
well, it's impossible to predict, but
like I honestly think like for us, I mean, who knows, we'll probably be working well past what our current average lifespan is, I think.
Like we could potentially be working and be well functioning at 80.
That's the goal.
Yeah,
that's the goal. So that's been the biggest challenge. I mean, aging itself, like probably over the last century or so, we've increased human lifespan quite significantly, mainly due to improved sanitation, antibiotics. We're really good at keeping people alive with heart problems now, but we just haven't had that same increase in health span over the same time period. So we might get an extra 20 years, but maybe only 1 or 2 of those are actually in good health. So we're sick for a longer proportion of our life. And part of the aging, most people in aging research is just trying to push that health span as far as we can. I personally think that when you do that, you're going to get an increase in lifespan. Some people think that there might be a theoretical maximum on lifespan. I can't see why there would be.
Okay, so now I think is a good time to ask, what actually is aging? How do we define aging, right? Because, well, I'll let you answer it, because there are stuff like the epigenetic clock and there's different stuff that you can explain better than me. But how would you answer that?
It's that's like also like it's a big question in the field, right? Like it's what do you classify as aging? How do you quantify it in an animal model? It comes down to really there's probably, I think there's 8 or 9 hallmarks that most people associate with aging. The problem is that no one of those hallmarks sort of acts on its own. So like some of those hallmarks might be genome instability. So the structure of the DNA and how that might change over time, whether that's due to mutations or just rearrangement of the genome itself. Epigenetic dysregulation is another. So this is what genes are turned on and what genes are turned off. So that's probably like a big one. I'll probably come back to that. I'll mention a few others. Some of the others are telomere attrition. So people have probably heard of telomeres before. So on the end of chromosomes, you basically have these protective caps. And the best way I've heard it explained is think about your shoelaces, and you've got that little plastic bit on the end of your shoelace to protect your shoelace from fraying and falling apart. Basically, our chromosomes have the same thing.
Okay.
But over time, through each cell division, they get shorter and shorter and shorter.
Yes.
So eventually they get to a point where they get into like the genes aren't functioning and you're just basically getting your chromosomes sort of falling apart, or you're just getting mutations through some of those genes that you need for that cell to be the type of cell that it should be.
Yeah.
Some other ones are sort of dysregulated nutrient sensing, so stuff that happens in metabolism. Mitochondrial dysfunction is a big one. To come back though, so there's a few others. Loss of proteostasis, which is how your body turns over the proteins and recycles itself. So as we age, our protein turnover. So if you misfold a protein in that cell, your body has a way of recognizing that and then sort of recycling it to use again, make the actual protein that it should be. And over time, we lose that ability a little bit.
Would it be fair to say, or is that too simple of a way to explain it then, to kind of summarize what you said? Is aging like a progressive deconstruction of our cells?
Basically, yeah. So if you want like I think I'll just try and think back to one of the seminars I gave, because I tried to give like a one sentence. It's the time-dependent accumulation of cellular damage.
Okay, yeah.
And ultimately like results in a loss of cellular function, which leads to tissue dysfunction sort of over time. To come back to epigenetic dysregulation, which is the big one that sort of David's on at the moment. So the way that our genome, and I'll try not to assume any biology knowledge for any listeners or anything like that.
Mine is very limited, so I will ask.
We'll go back to like the basic the dogma of biology, right? Is that you have DNA, and that's like the instructions for life, right? And that when you have a gene that's written in DNA, that gets sort of read into RNA, a messenger RNA molecule, and that gets turned into a protein. So DNA, RNA, protein, right? And then the proteins make up your cell, do everything that it needs to do. There's a reason why you very broadly, you have the same DNA in your liver cell as you do in your hair cell, right? But the genes that are turned on in the liver are telling that cell to be a liver cell. And the genes that are turning on in your hair cell are turning on the proteins like keratin or whatever to make your hair a hair cell.
Yep.
That's the way that the cell tells itself what genes to turn on or off is epigenetics, and it's way that the genome is actually organized in the nucleus. So you have these sort of big loops of DNA that are accessible by these enzymes that come in and read the genes that are meant to turn on.
Yep. So far, so good.
Yeah. So, and then over time, basically, you get this sort of, it's almost like a loosening of the genome where the bits that were tight before and then that were turned off and weren't read, they might start to get loose. So some of these genes are turning on. So you might get some of these genes that aren't meant to be switched on in the liver, they're switched on. So now this liver cell is like, well, I think I'm a liver cell, but now I'm starting to look a little bit more like an immune cell or something like that.
It's a weaker construction.
Yeah, you're getting like this loss of cellular identity, basically, sort of over time. And that sort of can be switched on by, like, that sort of thing can start to occur by mutations and like DNA breaks.
Cancer.
Yeah, so like some of these things, why these enzymes move around the genome is because of like a DNA break somewhere else. And it's like, I've got to go and fix this. When it's gone, all right, this gene's turned on. So, and it just starts this snowball. That's probably, if you want like the earliest the earliest marker that's going to affect all those other hallmarks of aging, at the moment, that seems probably like the best. The best case for targeting that to affect everything else.
And is that what they're looking for in the when they do the epigenetic clock reading of your cell? Yeah, that's what they're looking for, and how far along you are.
So with the epigenetic clock, you have you have a couple of different ways that the genes are turned on and off. One way is that they're just sort of packaged around histones that make up a chromosome. So you have basically proteins that bundle up the DNA. Another way to turn them on or off is with a methylation. So there's basically just a small chemical change to one of the bases, and that'll turn that gene off. I think loosely, I should check that, but I'm pretty sure it turns the gene off.
Yeah.
What this guy Steve Horvath, and people talk about the Horvath clock. Yeah. So he's a guy from I think California, and he sort of recognized through machine learning actually that he took like a whole heap of different tissues and sort of looked at them throughout age and looked where these methylation marks were. And he found that there's like these conserved, like a small number, if you think like the human genome is 3 billion letters long. So you don't want to count all the methylation marks, right? You just want to have the fewest amount that can tell you the most.
Yeah.
And he sort of like identified a certain number of these that no matter the tissue you look at or the cell type you look at, that methylation marks on these particular points actually correlate with biological aging.
Okay.
And a really, really good predictor. So we can use that now instead of just saying, because I mean, how else do you classify aging except for how many laps you've done around the sun?
Yeah.
Like that's really like before that is like, well, you know, how biologically old are you? It was the same as how chronologically old you were.
Yeah.
But now we can start to tease those two things apart because we know people age differently. Like you see it all the time.
So I think that might be something that the listener is curious about. First I'll ask an easy question. Is the epigenetic clock and the Horvath clock the same thing?
Horvath clock is a epigenetic clock.
Okay.
There's a couple.
Yeah.
He's probably the most famous one and the one people mostly use.
Heard of it. Yeah. Okay. Is there a way now for us, let's say here in Sydney, to be able to get a reading, like an epigenetic clock reading fairly easily and, you know, at a reasonable price, or is that difficult to get done now?
I'm not too sure about in Sydney or in Australia. We don't have our sequencing technologies to that level at the moment. There's probably only a couple of facilities in Australia that do methylation sequencing at the depth required to be able to see that. And as far as I'm aware, you need like a medical reason to have something like that done.
Oh, okay.
To the point I was saying before, with the reduction in cost in genetic sequencing, to the point where like I've got, you know, one on my lab bench, like I wouldn't be able to do something like that with that sequencer because the human genome is too big to do on that sort of thing. But that'll get to the point where people will start, I'm sure people will start up companies, start offering this sort of stuff.
I think I have seen it in the US.
Yeah, you
like
can.
My DNA or something like that.
You can get. I mean, 23andMe was the big sequencing company, but that was just looking for single point mutations in your genome. So they've been around a while. They might be offering a different product now. I'm not sure. I know back in the day, that's all they were sort of looking at. They just look at maybe like 200 or 250,000 single markers across the genome that are correlate with certain or associated with either diseases or ancestries or whatever, which I think was probably like their main market was giving people ancestry reports. Yeah. You know, and then maybe a little bit of health stuff, but whether that's actionable on some of those things, I think is a little bit questionable.
Yeah, I think what people mostly wanted out of that was to see where their heritage was from. Like, which I've got some Italian in me, some where.
So I had that done. Like when it first came out. Yeah, I had it done when maybe like really, really early on in the piece, where they were maybe like 6 months or you know, sort of a year into selling that.
Yeah.
And I was the same sort of thing. I was like, oh, this will be like, I'm interested in DNA. I'm interested in genetics. I'll sort of get my DNA sequence. And I got it back and it was like 98% British and Irish. And I'm like, okay, sweet. I could have saved my money.
That's pretty funny though. Yeah, I have, I guess when the epigenetic clock becomes more accessible for us, I do wonder what kind of usage we'll try and get it for. Because I have heard, you know, some places in Europe where they have a large amount of immigration and asylum seekers, people come in without any paperwork, and the government will have to treat adults and adolescents differently under law. Right. But then they're trying to because they don't know actually how old they are.
Right.
There's no paperwork.
Yeah, okay.
They're going to try and find out how,
Yeah, that's
what
an
law they
interesting
fall
use
under.
case that I haven't thought of, yeah.
And I'm sure there'll be others that we
Yeah.
haven't thought of at all. I mean, even in maybe like youth sport and stuff like that. I don't even know.
Yeah.
I feel like sometimes you watch these kids rugby games and there's some Islanders there.
Yeah,
like
it's
they're
always the
23.
way. Yeah, always end up lining up across from them going, how am I going to tackle this dude?
Yeah, but not necessarily that they're cheating, but you just never know, right?
Yeah.
Like, and if it's easy to do a test, then that might be something that comes up in the future.
Yeah.
All right, Tim, tell us a little bit about your specific study, and you had a paper done in bioassays.
Yeah,
Can you
yeah.
talk a little bit around that? Because I did have a read of it, and I have to admit there was a lot actually over my head.
Yeah.
So maybe, explain to us what you're doing and what your kind of niche is lining up to be in your study of aging.
Sure. So my specific study is looking at the biological effect of of DNA transposons and their relationship to aging. So
TEs, is that what it's
TEs,
referred to?
transposable elements. So it's worthwhile giving a big overview of transposable elements because I know when I started studying, I had no idea what they were.
Yeah.
But basically, when I spoke before about genome stability, and I spoke about the cell in your liver having the same DNA as the cell in your hair, that's not necessarily true. So for the most part, it is. You have the same DNA, but there are these elements called transposable elements that are the best way to describe them is jumping genes. So they're genes that can they reside in one specific spot in the genome, but they can either copy and paste themselves somewhere else, or they can cut and paste themselves somewhere else. So if they're doing that, wherever they paste can cause a mutation or have an effect based on where it's going, and we don't know always where it's going, right? So usually, when I spoke before about the DNA being tightly wound and you have these areas that are sort of closed up and inaccessible, they're usually regions of transposable elements. So the human genome has, in some estimates, 50 to 60% of the whole human genome is transposable elements.
Oh, wow.
Yeah.
And is the TEs, is this a fairly new finding that what they're doing, or is that
So it
while?
was originally discovered in the 50s. A lady named Barbara McClintock got a Nobel Prize for showing that she was working on corn, and she saw that there's some kernels that had different colors, right? And she was like, why do they have different colors? And she worked out that it was because these genes were jumping around and causing this sort of color change and sort of like a cancerous mutation or something like that.
Wow.
And so she got the Nobel Prize, and they're still largely, even in most biological science, they're usually still disregarded. And there's a couple of reasons for that. The first is that they're really, really hard to sequence. The second is that they, between each cell type, it might jump to a different spot. So you can't really tell if you're doing sequencing and you crush up a whole tissue and then you're sequencing it and it's like, well, I don't know which one's true or not, which one's contamination. So to try to define that resolution of where that's jumping is really hard, really hard.
Yeah.
And they're also really they're generally repetitive by nature, which in sequencing has been the hardest thing to do and when I say repetitive, I mean if you have A, T, G, and C as your DNA, if you have lots of A's and T's or whatever in a row, that's like a repetitive sequence. And that's really, really hard to do in sequencing and know where that came from. So that's why they're usually left out in genetic analysis. Like a lot of the time.
Just because it's so difficult to
really,
prove
really
it
difficult
in a scientific way. Yep.
But there is some improvements in that are coming at the moment in some of these new DNA sequences that are actually able to increase the resolution. So you'll end up hearing more about them, I'm sure. It's so from my point of view, these are turned on as we age, right? So as that genome unfolds and these repetitive elements get turned on, you can't stop it then, right? Because one turns on, it jumps over here. All right, now we're going to try and fix that mutation. This one turns on, and it takes off, right? There's some cool therapies targeting transposons itself that have got some pretty cool results in sort of animal models, mice and worms and that sort of thing.
Do you mean drugs that they're using? Or?
Yeah.
Yeah.
Yeah. So one of those transposons is called retrotransposons, because these are, a lot of people think they're viral leftovers in our genome from millennia ago. But we have so one of those, with the one that does the copy and paste, it actually uses an enzyme called reverse transcriptase. So if you go from DNA to RNA and then it's going back to DNA, that's like a reverse transcriptase step. And that's what like HIV does. Like HIV is the type of virus that does that. So there's some interesting stuff where people have used therapies that are like used in HIV treatment. Yeah. In this case, that are actually having some decent results in turning some of these elements off.
So that treatment can have some anti-aging effects.
Possibly, yeah. So that's like really early. That's like a few people around the world are sort of working on that at the moment. It's really, really interesting. Where my study comes into this, and I'm sure we can post the paper if people want to have a read of it later on.
I tried to give it to a couple of mates as well. They looked at it for a second and they were like, it's all over my head.
Too much.
I don't have too many science friends.
Yeah. So the other thing, I've got to give an explanation, obviously, of transposable elements, which are also like they're throughout all organisms. So transposases, which is the protein that sort of does that sort of thing, that's the most common protein throughout the whole of life. Like every organism, from bacteria, fungi, right through to complex eukaryotes.
Any organic material, basically.
Yeah, well, anything that's alive will likely to have transposons. So my study is looking at horizontal gene transfer of these transposons. So if you think about evolution, we think about passing genes from parent to offspring, right? And that's sort of the way. And then slow mutations give rise to new characteristics, and you eventually get speciation, and you sort of move through that way. There's this concept, and it's pretty well understood now, called horizontal gene transfer. And it's basically that, like, instead of passing genes just through your offspring, I'm going to give you a gene.
Yeah,
you're going to take it on to a completely different organism, right? And it's not that well studied in eukaryotes, so like humans or whatever, but in bacteria, they do this constantly. Like, it's the only way. There's no sexual recombination, so the only way for mutations in that genome is through gene transfer, right? So they're constantly passing genes in and out, right?
Do they do this with fruit and veg?
Oh, that would be like, you're probably thinking a little bit of genetically modified organisms sometimes, or is that?
Yeah, I'm not sure, but what I recently learned was that something like broccolini, we didn't really have that before.
Yeah, so that's probably more selective breeding. Okay. A lot of that sort of stuff is just choosing organisms based on characteristics and trying to push a certain direction. Yeah. Whereas this is more, left to chance, sort of like swapping in and out. So people have talked about antibiotic resistance, right? So that's usually happening on transposons or mobile genetic elements, where if you have an antibiotic resistance gene and you get some selective pressure, it's passed on to this organism now. So now this organism is resistant.
Oh,
right?
Yeah.
So what my study and what we think we've come across is that potentially some of these transposons from bacteria, we live with bacteria, right? Like, there's something like 10 or 1,000 times as much bacterial cells as human cells in our body, right? Yeah. So we live with a lot of different microbes.
Especially in our gut, right?
Yeah, in our gut, in our lungs, in our mouth, everywhere, on our skin. We think we've come across potentially evidence where these genes have left bacteria and jumped into a human cell. And as you can imagine, if that gene's jumping into, a gene that's essential for function for that cell, it might cause senescence and cause that cell to die off really quickly. It might cause DNA damage. It might start to turn on these other transposons in that cell. Yeah. Right. So the reason and why we think we've come across this and we haven't seen it before is that some of the pipelines that people use in genetic analysis and looking at sequencing data is, I've got a human tissue sample. I'm going to align that, like those DNA sequences that I get back, to the human genome. But the human genome doesn't have these bacterial sequences in it, so everything that doesn't match the human genome sort of gets thrown out. So now we're thinking, well, maybe instead of throwing that out, maybe we should look where else those sequences are coming from. and maybe some of these bacterial transposons are actually jumping across. And because it's not going into all of your cells in the same spot, so it might jump into one cell in one location and into another cell in another location. That's where I'm like talking about the resolution of the sequencing is you're not going to get like a consensus sequence to say, oh, I've got 10 copies. That's where it's jumped in every time.
It's random, right?
Yeah, it could be really, really random. So that's basically the essence of my PhD and what I'm looking at. So looking at, first of all, does this occur in human cells? Looking at existing data. So I'll be doing a lot of computer analysis on the existing data from aging data sets from all around the world that already exist, but also looking in the lab and seeing, can we see this happen in almost in real time?
To prove it, right?
To prove it, like to show that it happens. Okay, it happens in a tissue culture dish. All right, now let's take it to the next step and go to a complex organism and how it lives normally.
Yeah. So how do you see this type of study have a, like a practical effect, or it could potentially have a practical effect on us as like, your human species in the future? What are some of the flow-on effects that you would like to see from a study like this?
The first is just to see if it happens or not. And I think that there should be, it's always hard to say where new knowledge will take us, right? And a lot of people will push back and say, oh, you shouldn't do basic science unless like there's a research outcome that's applicable to the clinic or whatever. But we don't necessarily always know, right? If we're actually able to show this, that changes the whole game in biology. Like we already know that the definition of a species is really, really loose. And this just sort of adds complexity to that.
Yeah, I always think that when I see Shaquille O'Neal. I'm like, what? Are we the same species?
Yeah, that's fair. Maybe he's got more transposons or something. I don't know.
Something like that, yeah.
But when it comes to like some of the stuff that we've seen that other people have studied in this have shown, like in certain stomach cancers, there's an enrichment of these genes that have jumped across. So the first thing is understanding it. For sure. And I mean, it opens up a completely sort of new field of biology for people to start looking in this direction. The second, so once we know that it happens, how does it happen? What are the enzymes? What are the processes that cause this to happen? And then at that point, that's the point where you can start to develop therapeutics against those certain things.
Super early in the
early.
In the basin,
Yeah. Really, really early.
How much longer do you have of your PhD?
Probably 2.5 years or so. So usually, like in Australia, it's PhDs usually 3.5 years. But I know very few people that finish at 3.5. It's usually stretched on to 4.5. Yeah, more often than not. Yeah, it takes a while.
Yeah, but it sounds like something you're really passionate about, right? And working under someone like David Sinclair must be super exciting.
Yeah, especially at the moment. The aging field itself is really sort of taking off. So it's an exciting time to be in that field and like you said, sort of have contacts like David is always helpful.
It'll probably develop a lot even during this duration that you're doing your PhD, right? So that's
I mean, hopefully by the end of the year I've got some cool stuff to show, like it's one of those things that, I mean, maybe I might have nothing. You know? Like I might have nothing by the end of the year, or I might have something that's really, really exciting. So
okay, I've got to ask you something that I know David gets asked all the time as well. So it's a bit of a repetitive question, but what are some of the things that you're doing or that you would recommend for us to do now to live a longer life, but also not, we talked about that in the beginning, not necessarily just a longer life, but a healthier, happier life, right? Like one thing is to make it to 80, 90 or 100 years old. But if you're not functioning well at those ages, it doesn't matter really. Yeah. So what are some of the things that we can do now that you think that's mostly helpful for that?
I mean, it's going to be the typical stuff that, most of the listeners know. So it's exercise. Nutrition. It's sleep.
If we skip all those though, are there any drugs or anything like that we should be taking?
There's nothing that I'm taking. There's stuff that David's
You're not on resveratrol?
I'm not on resveratrol. I'm not on NMN. Our lab in Sydney works a lot with NMN though. We do some stuff with mice and rats with NMN. That's pretty cool. Yeah. But I'm
What's your take on that now? Like, what are you seeing from it? And
out of the lab.
Yeah, and why are you not taking that at the moment? Is that still very expensive?
I'm probably not taking it, probably just because I haven't asked my supervisor to get me some.
Is it expensive?
I'm not sure if it is at the moment because I don't work on it. Like I'm not the one ordering it, but I've seen it. Like I see it in the -30 freezer like all the time.
Okay.
So it's like a couple of like tubes of it.
Yeah.
But I think there's a company that we order it through that you can't buy it from.
Okay.
But I have heard, like, I mean, you can, apparently you can buy NMN online, but it's a supplement industry. So it's always really, really hard to know what you're getting. I know that someone has actually done a test on a product that said it was NMN and it had 0 NMN.
Oh, wow.
And the people are selling it online. So that's always the risk, right? With anything in the supplement industry, is knowing what you're getting is coming from a reputable source.
Yeah. I mean, we see that a lot with CBD oil now that, a lot of them are advertised because if you're going to have anywhere, stay clear of the law at least, like, you don't want any THC in it here in Australia. And, they've done some testing and some of them have like 1 to 2% of it in there, which is actually quite a lot.
Yeah. And I mean, no one's got the facilities at home to test this stuff themselves. No. So you've got to find, first of all, someone you trust and know. But the other thing is we don't really know yet what the optimal dose is for most people. I mean, David speaks about what he takes, and that's based on his years of research of what he's shown in the lab and sort of, I mean, a lot of that stuff comes from mice and rats and, I mean, there's only recently in the last few years been a few clinical trials in humans with some of these drugs. So the optimal dose range at what time point? I mean, am I too young to have an effect yet? Like, is 30, 32 too young yet? Or is it the right time to be taking that sort of stuff? Like, that's some of the questions that still need to be answered. Yeah. I mean, one of the studies that came out was sort of like combined with our lab and David's lab at Harvard, like 2 years ago was NMN in old mice and looking at exercise function, and they basically gave NMN in the drinking water to these mice when they were old, and they outran young mice on the treadmill, like crazy, right?
It's not safe.
But when they gave NMN to young mice and exercised them, it was no difference. No difference. So there's probably a point, or there's an age where you've got enough as it is. You've got enough NAD, which is what NMN does. NMN increases NAD. Yeah. So there's a point where you've probably got enough, and then as we age, that declines. I don't know what time point that is in the human lifespan.
Are there any other ways to increase NMD besides
NAD?
Yeah. NAD.
So there's a couple, so NMN and NR. So NMN is nicotinamide mononucleotide. And the other big one at the moment is NR, which is nicotinamide riboside. And they both, through sort of separate pathways, raise NAD levels. So NAD levels typically decline throughout age. I mean, anyone that's done sort of basic biochemistry would have heard NAD and remember back to really complex biochemistry diagrams and go, oh, geez, NAD, like this is too much. It's like something like 800 chemical reactions in the body use NAD, right? Yeah. But one of these enzymes that keeps the genome stable throughout life are called sirtuins, and sirtuins use NAD. So we think that the main reason for supplementing to increase NAD is acting on these sirtuins and giving them enough NAD to do their job to keep the genome stable.
That's awesome. That's a good explanation. But the only way that we know now is through, basically the only way we can increase it, right, is through the drug, NMN.
Through one of the precursors, usually, yeah. I know of some clinics that try to deliver NAD sort of intravenously, NAD itself. From my understanding, and again, like I don't work too much on NAD, but the molecule of NAD is typically thought to be too big to get inside the cell. So it's usually got to be a precursor that gets inside and then gets converted to NAD. Oh, okay. That's usually what people think. So
again, this is like super early days for this type of drug. The other one that's had a little bit more of a track record, I guess, is resveratrol. And as we discussed before the podcast, there was huge a couple of years back because we can get some resveratrol through red wine, and that's when this whole wave with drink red wine in the evening to live longer. What's your take on that? And explain a little bit kind of bit better than me. What happened through that stage? Because David Sinclair was quite involved
so
in
he
that
was
study
he
too.
was part of the lab, and if listeners remember, back in the day where there was this massive push for you've got to drink red wine, you'll live longer, and that came from this study, like these studies that showed that in model organisms, if you gave resveratrol to either yeast or worms, sometimes I think there might have been some mice studies early on, it extended lifespan, extended health span. And then people sort of, in media releases or whatever, it's like, oh, where can we find resveratrol? And it's like, well, it's in red wine. And it's like, oh, all you've got to do is drink red wine to live longer. And it's like, well, no, not necessarily.
All the newspapers jumped on that.
Yeah, obviously, that can be like media releases when you have a scientific paper can be a blessing and a curse, like if you can get some good publicity, but it can take off and absolutely sort of, kill your reputation in the scientific community. So that's the sort of thing that happened, is this huge pushback in the scientific community. Like, that dose that you're having in these model organisms is like barrels of red wine a day or something like that, It's crazy, right?
Yeah. Oh, wait, so you had to drink even more red wine
that's right.
to live longer to get the right
you just
dosage.
end up drowning yourself before you live longer.
I think the negative effects would outweigh the positive ones right there.
Yeah, I dare say. Yeah, I don't know if you'd have a liver left. So the part of that as well was people that sort of criticised the suggested mechanism for how resveratrol worked, and basically I think David speaks quite openly about this sort of time in his life, and it was like debilitating for him, and he was sort of down and out as a scientist. Couldn't get funding. Like really really hard, and that's something that's if you've got no funding in science, you're gone. Like you got to go and find another job, because you just can't support yourself. Like it's really, really, that's sort of the way science is, and that's how we've set it up. So he was super, super down and out, but resilient enough to go, no, like I know this mechanism is likely right. I've just got to do better experiments to show it. And I believe, I'm not too sure, I think we were speaking before, I'm not too sure if the paper is yet released, but I'm pretty sure it'll likely be out soon, where he does show the mechanism for how resveratrol extends lifespan and what its sort of mode of action is.
Yeah, so it's kind of the way I understand it is that it's almost gone full cycle. He originally released this information, and then it went through the media and hyped up, and then other scientists kind of debunked it, and then he lost his funding, lost a lot of his researchers, and then now it's kind of all the way back, and he's proven it in the
incredible story, right?
Yeah,
like as a
awesome.
scientist, like that's the stuff. I mean, I don't want to like lose all funding and be like down and out for a while, but to go from a molecule like that extends lifespan like very early on in really, really simple organisms such as yeast, and then sort of move that through all the way to humans, but also show the mechanism, like that's like complete story. Like people dream of that sort of stuff, And
often those stories in science takes decades.
Yeah, and it has for him. He, like David, worked on yeast in his PhD.
Yeah,
like it's back in like that's I think like mid to late 90s or something like that. So 20, 30 years ago.
Before I ask that actually, resveratrol now, is that something you can get in Australia as a drug over the counter, or
I think you can buy it as a supplement as far as I'm aware, but it's the same sort of thing, like where you're getting it from, what's the dose, like these sort of guidelines aren't really out there. Again, like it's like none of these things, either NMN or resveratrol, I don't take any of them. Again, like would I? Maybe. Yeah.
In the future.
I don't at the moment. It's just not huge. Like I'm super, super interested in aging, but like it's like I'm more interested in getting through. I'm still young, and I'm just like trying to get through my work. Like,
The way I've understood it from listening to David on podcasts is that resveratrol does have some major benefits when you look at aging and longevity, but it also has some downsides through, if you're a high level athlete or you train at high intensity, it can have some negative effect on that. So it depends what you're.
Yeah, and a lot of these drugs will be quite similar. So I think there was a study, and I can't remember if it was NMN or NR. I think it might have been NR that showed like a decrease in aerobic capacity.
Yeah.
I will say there's no such thing as a free lunch. So you have to look at all these sort of things, and it's still early days to know everything that's happening, and that's why we need more funding, clinical trials, get this stuff through. One of the big things, and we work really closely with a fertility lab at UNSW, and age-related fertility loss is like one of the first things that sort of happens, and it's like, again, across all organisms, it's conserved that as you age, you lose the ability to reproduce. And obviously that's like a huge thing for society, right? Like, you know,
you're getting kids later and later, right?
Yeah, but to be able to push that later and later and have that choice to be able to have kids later and later might take the pressure off people trying to choose career over kids, or, you know, having to make those sort of tough decisions. And we just, out of our lab and the fertility lab at UNSW last week, released a paper that showed men recovered reproduction in aged mice.
Wow.
Yeah.
It's so new, hey? Like, you guys are releasing this stuff.
So,
now.
like, you know, treating these mice with NMN in their drinking water, and like versus controls where you're getting basically sort of no embryos. Yeah. In this one, you're getting many more, like recovering it really well.
I say it all the time, but we live in an incredible decade where, you know, where we're at with science, it's just mind blowing, and where we're going to be in 10, 20 years. Yeah. I'm trusting that all my issues health-wise is going to be resolved by science.
Just forget about everything.
Just like, go hard in the paint.
Worry about it later.
Nah, that's not quite true, actually. I live fairly healthy. But Tim, what I wanted to ask just before there was, where do you see yourself within science, like in the future? Is there something that you're going to pursue? You obviously have a career already as a firefighter, but the science part must be super exciting as well. Yeah,
it's definitely where the passion lies at the moment. So I sort of studied late. I didn't study straight out of school. Yeah. I got into the fire brigade early, and it was sort of maybe 6 or 7 years into the fire brigade, and I was sort of getting not necessarily itchy feet, but my mind was wandering, and, you know, I was like, I'm trying to find something else, you know. So it was a little bit of personal training, doing some labouring on the side, you know, like some building, some landscaping, and then I was like, there's got to be something else. Like constantly
choice between being a scientist or like PT or labourer. It's not pretty easy.
But I was always interested in science, as a kid or like through high school or whatever. But I just never sort of thought about it, and then I was like, oh, maybe I should go back to uni and do a degree. And I was actually tossing up either between something sort of health related, because obviously I was passionate about that just through training and that sort of thing. Or I was going to go the point of like physics and like astronomy. Yeah. I was like, okay, like, you know, the universe. And like, I was like, the other books that I read, like was those sort of things. And I was like, no, I should probably do health, because it's probably got more of a future in it, you know. Like, I mean, obviously, there's jobs for physicists, but not as many as there are in sort of health science or medical science. So I did like an undergrad in genetics and biochem. And then I finished that, and I was like, well, like the next step is to do an honours degree to get into a PhD program. And that was where I sort of approached Lindsay and David at the Laboratory for Aging Research. And I was like, at that point, I hadn't really looked too much into aging research, but I was like, oh, this is new, this is interesting. Yeah, like it's like on the cusp, like super, super keen. So I got into the lab and like, loved it, enjoy the lab. Enjoy the work. It's like, you know, really, really, really passionate about transposons, which is like, I never thought I'd say, and aging research in general. So sort of decided to stay on and do my PhD with the goal of eventually getting out of the fire brigade and going into that full time. I'm super fortunate to be able to have the ability to do both at the moment, because, you know, it doesn't often happen that way. Most of the guys that I work with in the lab, that's sort of all they do, whereas like the fire brigade sort of affords me that opportunity, especially because I'm running my own research, that I can sort of adjust my research around my work schedule. So that's, I'll try to hold on to the fire brigade for as long as I can, obviously, but I think eventually it'll get to the point where I've got to make a decision, possibly to go overseas and take a postdoc job over there or something like that, you know, which is why I started it in the first place. So it'll be hard when it comes that time to make that decision, because, as I said, I've always been a firefighter, like since, you know, 19 years old. So people are, oh, what do you do? I'm a firefighter. So when it comes that point where I've got to go, oh shit, I'm a scientist. It's like I don't know, like I'm not ready for that now, but like, I know that that's coming, like eventually. So,
but you still got a fair few years left with your PhD as well, though.
So it's not, I don't have to make that decision anytime soon, thankfully.
It sounds like you have, and to be honest, it sounds like you have an enormous workload, really, working as a firey, having shift work, and then also doing your research. How do you balance all that throughout the week?
It can be tough. It can be tough, and I'm sure my girlfriend can attest to that. It probably affects her more than it affects me.
Yeah.
But yeah, I mean, like, so it's to the point where, you know, I finish a shift and then I'm going to the lab, you know, and then I'm coming home and sort of doing a little bit of computer work or whatever. So it's quite constant.
When do you sleep?
Yeah, at the moment we just got a new puppy, so that's even less
wow.
now. Like,
Oh, really?
Yeah.
What'd you get?
We got a Hungarian Vizsla.
Oh, wow.
Yeah. So she's like super active, and she's not sleeping through the nights, and so like the little sleep I got is like even less now. So
wow.
but that'll get better. It's one thing that like I should probably focus on more, but I find it really hard to sort of switch off, especially at home when I can do sort of work on my PhD on my computer.
Yeah.
It's like, oh, okay, I should at least be reading a paper. So I at least get to the point where I try to do like one productive thing every day, like that's going to contribute to that end goal.
To the PhD.
To the PhD. So even if I recently sort of I went away for the weekend and like on the plane, like I read 2 papers. And I was like, okay. And then I was like, and then I can switch off and I can be like, okay, I've done something constructive today.
Yeah.
Now I can like afford to switch off.
Do you think of that as, you know, every day for 7 days throughout the week, or do you try and take a day off?
I try to do one thing a day.
Okay.
At least one thing a day that's going to be constructive to that end goal. But that can be super small as well, you know.
What's a small thing?
Read a paper.
Okay. Wow.
Yeah.
Okay. So, because I did want to ask you what are some of the specifics that you do when you research a PhD? Because I never did that. I did my degree at uni, but I think that's a lot easier than researching a PhD. You're a lot more independent. You control your own time, and you got to get the work done.
Yeah.
So obviously one thing that you mentioned is reading papers.
Yeah.
What are some of the other things, and like how does it look like the actual
like how
work?
does my day look?
Yeah.
So the first year of a PhD is basically you're sort of generating your project, and that's through looking at papers, what's known in the field, what's not known.
You got to come up with something new.
Come up with a research question that you want to answer and make sure that no one else has already answered it. And that's actually a super difficult thing to do because there's a lot of people that have done a lot of stuff before. So like the first year, and that's sort of what culminated in the paper that we submitted recently to Bioessay. So that was basically a literature review, but it was a little bit of a twist in it in that it was a hypothesis review. Yeah. So like you have a review and then you generate a hypothesis. And that's basically what you do. Like that's what you sort of need to do in the first year of your PhD. Generate, look at the literature. What's the review of how it is? Okay, what's my hypothesis and how am I going to test it? And then we start to work out, all right, let's start to plan some experiments on how we'll test it.
It's kind of science, basics of science, right?
Basics of science. Yeah. So, I mean, for today, for example, I'll go into the lab and I'm actually doing some stuff with the fertility group, doing some sequencing. So I've got some DNA samples. Well, I actually had some oocyte samples, which is the egg samples. And we're going to sequence the DNA of those oocytes. So basically when I go in today, I'm prepping that DNA and then going to run it on the sequencer.
So that'll in a lab coat and everything?
In a lab coat, yeah, with a pipette like in my hand constantly. So doing that sort of thing, running that. As I said, I do a lot of stuff on the computer as well. So once I get all those sequences, then it's like computer-based analysis, and it's like what we call bioinformatics, which is basically data science for genetics.
Okay.
So looking at, because you're generating gigabytes of data, right? Sometimes more. And then looking at, okay, how do we tease this apart now? What's actually going on?
So it's gigabytes of data, but it's not, I'm used to working with images and movie files, which gets big pretty quickly. But yours is just numbers.
ATs, Gs, and Cs. That's it. Basically.
AT, Cs, and Gs, they're the letters for the DNA sequence.
DNA. Yeah.
Cool. We should probably explain that little basic that I learned, a couple of days ago. Researching for this. Yeah, awesome.
So all DNA is like AT, G, and C.
Just sequences of that.
Sequences,
right? Yeah. Amazing. All right. I think we definitely, we covered a bit today, but I think this is kind of a never-ending subject. It'd be awesome to get you back in maybe a year or 2 years
when the
studies is going and what else is new in this field. What is actually some of the it might be one of my last questions. What is some of the new things that you've seen recently that's kind of, is there anything that's turning some of the stuff that you're working on its head and be like, it's brand new? Or
the stuff that I'm seeing at the moment is in sequencing, in these new sequences that are coming out, and the ability to do so much more with so much less.
Yeah.
Either that being like so much less sample or just less cost.
Yeah.
So that's really, really exploding at the moment. And, with that drop in cost, like that just puts so many more people in the game. So
it's a big thing, right?
Huge. It's like giving everyone a mobile phone.
Yeah, well, yeah.
Like you just change everything, right?
It's a perfect example, right?
Yeah.
I think I've talked about this before on the podcast, but there is having a mobile phone is quite powerful because you have so much information in the palm of your hand.
Yeah.
But up until fairly recently, that's been pretty much only the wealthier part of the world, if you're thinking globally, that's been able to have that phone and run a business through their phone and stuff.
Yeah.
But now that cost is coming down. Google has their Google-Fi, internet. Now it's their Wi-Fi. Basically, they're trying to make that available globally.
Yeah,
and it's free to
SpaceX putting satellites up for global internet.
All that stuff. Yeah, it's so much
that's
cheaper.
That's a democratization of some of those things, and that's happening in science as well. Usually only ever exceeds the potential of where we're currently at. So that's, I think that's probably the most exciting places. The stuff that we're starting to learn using these newer DNA technologies are things that we never saw in the past because the technology just didn't have the capabilities to look for it.
Yeah.
So it's like, well, we didn't think it was there because the technology wasn't there to look for it. So that's becoming really, there'll be lots more in epigenetics, in these modifications of DNA. There's a huge field about to explode in looking at modifications of RNA and how that affects biology.
Yeah.
Which is just people have only dipped their toe into that at the moment. That's honestly going to explode. And that's another level of complexity to get your head around. It's not just ATs, Gs and Cs and modifications. So there's a lot happening.
It's an incredible wave that you're part of now, though, because the science community is global, right? There's people starting this in Europe, in the US, in South America, everywhere.
And having access to those people, I actually find one of the best resources in the last 6 months, which I only jumped on recently, was Twitter.
Yeah. Oh, wow.
Yeah.
Okay.
So people talk about academic Twitter, which is this, sort of sub of field of Twitter where it's all these academics posting papers, posting tips, easy to reach out. When else could you reach out to professors in New York or wherever to get a tip on something you're doing that that they've done? So that again, that opens up anything. And I get heaps of papers through Twitter that I would never have get through through a Google search or set up. So I scroll through Twitter. My bookmarks in Twitter, just keep adding to them. The papers that I'm likely to never read. That's usually where I go to when I write one paper. It's go to my bookmarks, pick one of those.
What's a good science account to follow on Twitter for more the general public?
General public. That's hard. David's pretty good on Twitter.
Oh yeah, yeah.
David's pretty active these days on Twitter. Basic science. I don't know. A lot of the guys I follow are transposon guys at the moment.
Way over our head. One amazing resource that I have discovered. Well, it's part of YouTube. I mean, I've been on YouTube for years, but an amazing thing that started to happen over the last couple of years on YouTube is these universities putting out full, complete lectures.
Yeah.
MIT, Harvard, and you can watch the whole thing online for free.
You can do a full degree if you're not worried about getting the certificate.
Yeah.
If you just want the information, you can do a full. If you're motivated enough, you can do a full undergrad degree for free online from anywhere in the world.
Amazing.
Anything you want to learn these days, a lot of the time is online. The information is there. It's harder. It's becoming harder to filter through good information. The science stuff is generally pretty good. Just generally, stay away from the anti-vaxxers and all that sort of stuff. Yeah. Generally, it's pretty good.
Yeah. It's easier to filter through the science, I think, because
if you stick to the right people and the right institutions and stuff like that, you'll find the right information.
Yeah. Does the UNSW put anything out on for free, or
I'm not too sure if they do. I haven't seen anything. Every now and then they might release a small video or something like that if someone has released a paper, but I don't think they do any courses for free.
They got to get on that.
Yeah,
it's so good.
It's huge.
Yeah. Awesome.
Huge.
All right, Tim, thanks a lot. That was really good. And we'll get you on in a couple of years and see how much we've aged.
Thanks.
Or how
Thanks
little
for
we've
having
aged.
me. Cheers.
Thanks.